Our third presenter, Olmeda Biosciences, Shreya. Excellent.
Good afternoon. Today, millions of patients worldwide face the unmet need for personalized eye care. My name is Shreya Siribolu, and I'm a medical consultant for Olmeda Biosciences. Our company is built on technology with the potential to revolutionize personalized eye care through the use of ocular liquid biopsies. To illustrate the potential of our liquid biopsy platform, let's look at uveal melanoma, which is the most common intraocular cancer, with a market size of $835 billion. There are about 3,000 new patients diagnosed yearly, and 60 to 70,000 live in the US. And about half of these patients will soon develop metastatic cancer in their lifetime. An important aspect of managing the care of these patients is to determine their risk for metastatic cancer. Castle Biosciences offers a one-time tissue biopsy test that can be used to stratify patients based on their metastatic risk. So what is the advantage of a liquid biopsy? With liquid biopsies, we can simply sample fluid called the aqueous humor from the front of the eye, offering a much less invasive method of biopsying the eye than tissue biopsy of the cancer. What sets our ocular liquid biopsy apart is its exceptional safety and minimal invasiveness, allowing for serial sample collection. This enables the opportunity to go further beyond than anything that's currently offered in the market, allowing us to monitor the patient's cancer at a molecular level in real time. This end-to-end monitoring can inform us on critical questions such as, what is the best treatment? Is the treatment working? And when should we stop the treatment? Our company is a CHLA spinoff founded by Dr. Jess Berry and Dr. Liya. We started working with retinoblastoma, and we currently have a CLIA LDT servicing 300 patients and accepting samples from 9 centers across US. For uveal melanoma, we're in the process of licensing existing intellectual property from CHLA. Beyond the realm of eye cancer, our innovative technology holds immense potential for glaucoma and other intraocular diseases with even larger markets. Here you'll find our 3-year timeline. Our primary objective is to secure $3 million in funding to propel the advancement of our technology, with an additional $5 million earmarked for the support of our vital clinical studies. In conclusion, I would like to underscore the paramount importance of our mission in addressing the unmet needs for personalized eye care. Our technology offers a novel, innovative solution that can truly revolutionize this field. We hope to have your support in creating a lasting impact on ocular health. With that, I would now like to introduce Dr. Liya, the clinical co-founder of Olmeda Biosciences, to answer any questions you may have.
My name is Liya, and I am the scientific co-founder of Olmeda Biosciences. And at the same time, I am the faculty of USC Michelson Center. And thank you very much, Scott, to recruit me 9 years ago and open the door for leading this work here. And I'm happy to take any questions.
This is an exciting product. Do you have any information, sensitivity, specificity, or ROC curves, diagnostic information?
Yes, we are working on that. But as a rare disease, it is super hard to develop these kind of numbers. But so far, we have tested 60 patients. And with a bigger validation set, we should be able to provide the number in this quarter.
Sorry, it might be mentioned that I didn't catch. What are you? It's a liquid biopsy, but specifically, what are you testing?
This is a liquid biopsy, but this is not a common blood-based liquid biopsy. So instead of collecting the blood streams,
I get that part. Yeah, is it DNA? Is it protein? What? Yeah.
This is. Every disease is different, and the purpose of the application is different for every stage. So, for example, for retinoblastoma, to diagnose the disease, we need DNAs for the genomic testing. But to evaluate whether or not this disease has a ocular salvage risk, then we will need RNAs. And for the treatment response or minimal residual diseases, we will need actually extracellular vesicles. So you will see this is not a simple genomic testing like current blood liquid biopsy are offering. This is with minimum residue, with minimum liquid, providing different omics knowledges for different applications.
And how much liquid do you, how much liquid do you need?
Clinically, we can collect up to 100 microliter from kids and up to 150 microliter from adult eyes, but we don't need that much for every testing. So we have the opportunity to minimize the liquid extraction volume and keep the safety maximum.
Sorry, I'm just have a lot of questions. What is the, how early can you catch the disease? And if it's an early stage, what's the prognosis for that early stage?
Currently, we have 3 IPs, but none of that covered that question. This is a very significant question. We have launched this study like retrospectively. Started with very late, very advanced disease, but now we are pushing it back earlier and earlier. And for retinoblastoma, we have the very first prospective clinical trials launched in CHLA. And with that, we can catch up early as stage 1, like stage A diseases. But for the uveal melanoma, we only have 1 established IP yet. So we are hopefully to open the clinical trials to answer that question.
Thank you.
Could I ask one more question to specify? So this is more of a diagnostic technique than an actual technology per a specific disease. So you can use it for 4 or 5 different retinal diseases.
Exactly,
Okay.
exactly.
Nice presentation for both of you. I had a question regarding your sort of market plan. I mean, because I can easily see that as you expand your IP, it can be more of license, and the technology, the procedures themselves can be utilized by physicians nationally.
Exactly, exactly. That is also our unique advantage on the go-to-market strategy, because so far all the IPs are generated based for the eye cancers, and eye cancer ocular oncologist is a very tight group. Actually, in US, there are only like not more than 100 ocular oncologists, and all participate in a very tight group. And our medical co-founder, Dr. Jessie Berry, is the vice president of International Society of Ocular Oncologists. So she gets to know like 300 elites on the planet. So our strategy is to keep our product, keep our knowledge as open as possible. Everybody are welcome to get our SOPs, get our knowledges, but to be honest, it's not that easy for them to develop any test specifically. So it is our job, like to not only to build up the knowledge for the society, but also provide a solution for them when they need it.
Thank you.
You're welcome. I know this is new, but this is very exciting. Thank you.